Hi, my name is Christine Cash, and this is my Animal Science 313 Physiology project. I'm going to talk about a collapse condition that is found to be genetic in Labrador Retrievers. Exercise-induced collapse is a condition that affects dogs during exercise. It is a heritable condition and is caused by a mutation of the dynamin-1 allele. This causes hind limb incoordination and also causes non-painful flaccid paraparesis. Dogs do recover from the episode after about 15 to 30 minutes of rest, but sometimes dogs can die from this. After the first episode occurs, dogs cannot participate in that strenuous activity again. This is known as a trigger activity. Exercise-induced collapse is most commonly found in Labrador Retrievers. This condition is found in those that are used as in conformation shows or just as pets. I cannot find why these populations were more likely to have this condition. Researchers also pulled data from Labradors that participate in hunt and field trials and that are service dogs. The mutation condition was also found in a few related breeds, such as Chesapeake Bay Retrievers, Flat-coated Retrievers, Boykin Spaniels, mixed breeds that may have Labrador in them, and a few other unrelated breeds. The dynamin allele is the allele that is responsible for the mutation that causes exercise-induced collapse. The dynamin allele is important in synaptic vesicle recycling at nerve terminals during high neurological stimulation or exercise. Miner et al. Determined that the DNM1 mutation is consistent with an autosomal recessive mode of inheritance. Researchers used blood and buccal swabs to determine if the dogs do have this DNM1 mutation. Dogs that tested homozygous for the mutation would experience several collapse episodes with excitement or exercise. Those that tested heterozygous or EN for the mutation do not have an increased risk of collapse. The homozygous genotype was found to be highest in Labrador Retrievers that are used for conformation shows at 13.6%. The genotype was found lowest in Labradors used as service dogs at 1.8%. On average, labs with the mutation will experience their first collapse episode before 4 years of age. This is a table that shows the prevalence of the collapse phenotype in Labrador Retrievers with different DNM1 genotypes. As you can see, even if they have a genotype that would show that they are negative for collapse, they can still have collapse episodes. And as you can see, those that do have the homozygous for the mutation do have a very increased risk of this collapse condition. There is other research being done that is related to this field. Research being done by Parker et al. Is mapping the genome of the dog. They say that the dog and human diseases are very similar, and if we can understand and study diseases in dogs and understand the dog's genome, it will help researchers when they go to study human genome and other genetic diseases. And even more research is being done by Eckenstein et al., and this research is studying idiopathic epilepsy in dogs. It is also found in humans and it's a very debilitating disease. If researchers can identify the genetic cause in dogs, then because of similarities between dog and human diseases, they might be able to find the genetic cause in humans as well. In conclusion, research is being done to try to find genetic causes for diseases in dogs that could help find the causes of similar diseases in humans. Exercise-induced collapse is a condition that affects Labrador Retrievers and other breeds related to it. There's a very small chance that a dog can die from an episode and generally recover quickly after an episode occurs. Care needs to be taken to avoid trigger activities with dogs who suffer from this mutation. This is my bibliography. Thank you for listening and have a good day.